This house, these are here.
Okay, good morning. Let's have a class. We will finish the lecture 4 slides, and then move on to the next bunch of slides. Will be the last session for our exam 1, and the exam 1 will be next week Thursday. Okay, and the next week Tuesday we'll do a very rough review, and the practice exam questions I'll put on the campus Thursday. Okay, then you have a whole weekend to do the review by yourself. So we're going to talk about Pili or Pili. Very briefly, without me spending too much time, let's say I have a bacteria. Sometimes have these expenditure right there, and these like a hair shape, and hair shape very thin. These are called pili. And actually the pili, you cannot use naked eye to see it. It's very small, tiny. You need to rely on transmission electron microscope. It's very, very thin. Now sometimes there is some of the structure very similar to pili, which is relatively long. This is called sex pili. And the sex pili we already mentioned is the function is for conjugation. And this is one of the genetic information transferring methods. And another two is transformation and transduction. Transfer methods. So just want to let you know, rely on sex pili. Now, there are some of the research also say the pili have a little bit very limited moving, or we say motility capability. But it is not that obvious, very limited. Okay? So we just briefly mention here. So we really want to spend time talk about this guy, is flagella. Now we already mentioned the flagella composed by a protein called the flagellate. So it is actually a protein. And it is very soft. So we use a special stain called flagella stain. And the flagella stain we already mentioned, need to use tannic acids to do a final coating. So it will be showing up. Now, what is a function for flagella? There is actually two. Number one is very important. It's called true motility. Bacteria rely on flagella to do the movement. Okay, that's true motility, or physical movement. The second thing you also need to know, the flagella is an antigen. For example, E. coli 0157H7. This H is flagella. So it is a flagella antigen could be using for testing microorganisms. For example, testing E. coli. And we gave them a serological number is 7. Now, based on the arrangement of the flagella, there is a 4 different categories. The flagella could be only at the end. This is called monotrichos. Monotrichos. What does the trichos means? T-O-I-C-H-O-U-S, from Greek word, this means hair. So that's why they describe the flagella, looks like a hair. Monotrichos, only one tail at the end. That means the bacteria have a very limited motility capability. Example will be Pseudomonas. And we have a Pseudomonas fluorescens, Pseudomonas aeruginosa, which is could be a sporage bacteria. And for example, E. coli. they have a very limited motility capability. And we will test in the lab, and you can see it. This is the first category. Second category, well, they could have a flagella at each end. You look at the picture. One flagella at each end. That is called Amphitrichos. Amphitrichos are a good example. It is Campylobacter jejuni. And we mentioned it is a major pathogen in the chicken meats. Limited, strong motility. So there's some, I will say some motility capability. I will be write this use red color, like this. The third category, that means at each end there is a couple flagella there. That is called the Lophotrichos. A good example is Ludosphaerum luberum. This bacteria is viral shaped. I used to have it, but do not have it anymore. It's there. Ludosphaerum luberum. Lophotrichos, couple flagella at one end. The last one we mentioned, the flagella is covered the whole surface and it is everywhere. That's called Petritrichos. And a good example is Proteus vulgaris, which indicates it has a very strong motility. And we mentioned about that, how strong it is. There is no colony forming even. No colony is forming in agar, on agar. So basically, for this bacteria from the agar, you will see it looks like this. No agar, no colony forming, we call it swan. You will see a bunch of like this, a smear. For Proteus vulgaris. Okay, so that's a very important for different category of talk about the arrangement, different type of the flagella. Now, how we determine a bacteria have a strong motility or not strong motility? We using a semi-solid agar. Some agar like this, they containing 0.5% agar. So we call it a semi-solid. If the bacteria has limited motility, when you do stab inoculation back and forth, it will be accumulated only in the middle. This means no motility. Or very strong motility, or very limited. If you see another bacteria which has a movement or the crowding or turbidity cover the whole semi-solid media, this means you have a strong motility. Okay, we will do this testing in the lab, a semi-solid agar. We'll do that. So that's for flagella. Now, real quick, for the spiral sheets, it's a special bacteria. For example, Treponema palladium. This is a pathogen called syphilis, and you need to use dark field microscope to do the observation. Instead of they have a flagella, they have an endoflagella. So endoflagella, very much like this. Asexual filaments, we call it endoflagella. Asexual filaments, we call Endoflagellum. Okay, that's for spiraceous. It's very special bacteria. Because bacteria has flagella, they could do a real movement. They could censor the chemicals surrounding them. There could be some sugar attracted. There could be something is stress information they have to repel, they have to flee away. Therefore, come out with chemotaxis. So what this means, if you look at this chemotaxis, this is basically means round trip for bacteria. If there is nutrition, the bacteria will go there. If there is a stress information, the bacteria will be repelled and flee away. That's called chemotaxis. So you can see the picture here. If there is a capillary tube has no traction, they flee away. If there's something, they go there. Okay, chemotaxis, the key points is a round trip. Could be go there and leave away, back and forth. Like you go to the trip from the Labor Day. You go Florida, Disneyland, you have to back. You have to buy a round trip tickets. The same thing for the bacteria. Okay. Last one, endospore. Very quick, we want to mention this. Endospore. We already did in the lab for endospore step. So just something we want to emphasize here, a couple of items. Number one, we mentioned 2 bacterial species have endospore, which is Bacillus and Clostridium. We will mention Clostridium very detailed in the examination too. It is an endospore forming streak anaerobic bacteria. Not only they have endospores, they cannot grow in the presence of oxygen. And the principle we will talk real quickly. So Bacillus in the lab, we already see Bacillus subtilis and Bacillus cereus. Bacillus basically happens is in the rice. So if you buy a rice from the Panda Express, some of the Chinese restaurants in Morgantown, then you left them on the bench, they didn't eat about 2 weeks later on, you see the molds there, that could be have endospore, which is Bacillus. So don't eat it. And don't microwave it either. The microwaving heat will not kill endospore. Okay, that's Bacillus. Clostridium. Clostridium, we will talk about in very much detail because it's encoding Clostridium tetani. Remember DTaP vaccine, diphtheria, tetanus, pertussis, Clostridium tetani is for that. Clostridium difficile. This will cause colitis, water diarrhea. It could be Clostridium botulism or botulinum, cause botulism. It could be Clostridium perfringens, which is caused gas gangrene. So we will mention these. But I just want to tell you there's a couple of species for the Clostridiums, they're all going to form an endospore. Okay, what going to be the endospore looks like? And the multiple layer, we already mentioned. In the middle, you can see it, I'm going to draw it, it's going to call the core area. Okay, then you see there is a inner cell membrane. Then you have another layer, which is inner cell wall. Then you have a layer right here. This is called outer membrane. Then you have another layer, which is called cortex. Then you have another layer, which is outer cell wall. Then you have another layer, which is called explorin. Multi-layer. So we call rigid system and a very tough system. Okay. And the bacteria in those spores we mentioned, it is a protection structure. Especially for stress environment. Let's say heat is one of them, and the UV light will be one of that. Sometimes acid, sometimes desiccation, which means very dry, no water. And relate to the water activity concept that we're going to mention real quick. But why the endospore can survive, to help bacteria survive? We mentioned in the core area, there is two important items. Number one, BPA, CA, calcium dipropanic acids, dipropanic acid. Second, small soluble DNA binding protein. Right in the middle of the core area, they can help bacteria in those spores to survive in the stress environments. If the bacteria forming in those spores, we call it sporulation. That sporulation is a very dormant status. It could be a couple of weeks, years, or maybe 100 years, or more than that, and beyond. When the condition changes, the endospore formation could be go back to the vegetative cell. So we call it a germination. So the germination happened when the condition changes from the endospore to vegetative cells. And we also mentioned the location of the endospore, it is important. It could be right in the middle, central, it could be terminal, and it could be subterminal. And the location is genetic dependent. So for example, Clostridium, that could be terminal, and the Bacillus could be subterminal. But you have to know the endospore formation is not genetic. And we talk about that the mother cell have endospore does not mean automatically the daughter cell will have endospore, except the daughter cell in the same stress environment. Okay, so that's endospore. I want to tell you, we'll give you an example when we move to talk about the Clostridium perfringens. In our life, the endospore, the big risk is for low temperature, long time cooking to make a soup. It will have a bigger risk. And I'll give you an example when we talk about that. Okay? So that is for the endospore. Okay, so we finished talking about this section, spending a lot of time, and the key here is the cell wall structure. These two pictures are important, and we will have a practice exam, let you guys to do the practice, to do the label. And in the real exam, we also going to test that. So that's the key things for that information, okay? So we finished this section, we're going to move on to very important lecture 7, it will be lecture 5, I would say, is for microbiome bacterial growth and nutrition, and then we're going to talk about the medium. So that's another big topic. So we want to move all these off and talk in detail about the bacterial growth. And lots of the things we could talk. By the way, some of you has already had the question. Lots of the chemicals I wrote on the blackboard does not mean they're going to be tested in the exam. The exam will be focused on the slides and also the practice exam. And but all these notes, which has helped you to understanding those, so you can take a look. But certainly we will not test you the structure, because it's not a biochemistry class or an organic chemistry class. When we move on to examination 3, we have extra points for you to know some of the structure. We'll mention by the time, okay? So generally it's not required, but we will just tell you what it looks like. So the first question in this section is microbial growth. Okay, microbial growth, it's very special, I will say. It's not like a human being growth. It's like a development from baby to adolescence to adults and become older, lots of topics. This is a very simple growth. It's called a binary fission. So what means a binary fission? Let's say you have a bacterial cell, like this. Okay, that's a very simple bacterial cell. Intersection is the genetic information, DNA or RNA. When they generate a daughter cell, they first of all is enlarged, become a little bit large, and then they will have a cell wall to break them. So what happens is this wall, a block forming, and then the genetic information ideally will be equally separated into two daughter cells, and finally you will see each are carry on a same copy of the DNA information. This is 1 becomes 2. That's called binary diffusion. And ideally, the genetic information will be exactly the same from mother cell to daughter cell, but it never happens because there is a mutation. The same thing, you looks like your parents, but not never the same, because mutation happens the same for the bacteria. Okay? How about here? This guy gonna continue to do, this guy gonna continue to do. So become another cell. Okay? Like this. Here will be another cell. Exactly the same. This is another cell, this is another cell. So we draw it. Then becomes what? Becomes 4. And on and on and on. So what happened? Binary diffusion will generate 2 times n bacterial cells. Okay, that n indication generation. How many generations it has? Therefore, we come out with a concept called generation time. Generation time, said very simple, is time required for bacterial cell to doubling the original number. That's called generation time. Some of the bacteria, the generation time is very short. For example, E. coli. E. coli only need averagely 20 minutes. That's the reason E. coli we can grow overnight, and tomorrow in the lab you can use it. And lots of bacteria, because the generation time is very short, which means 20 minutes 1 become 2, another 20 minutes 2 become 4, 4 become 8, on and on. And 2 times the n. But some of the bacteria, the generation time is very slow. example, Mycobacteria tuberculosis. Tuberculosis. Let's say Mycobacteria tuberculosis. This need maybe 24, 48 hours, even longer than that also. 72 hours sometimes. Therefore, we can rely on to cultivate Mycobacteria tuberculosis to do the identification. So we need what? Acid-fast stain to do a preliminary identification. Is that right? That's what we mentioned. Because the generation time is very long. At the end of the day, when you really get isolation from the Mycobacteria tuberculosis, you need to put in a transfer medium and go to a transfer lab, transfer reference lab, we call it. And that took about a week to diagnose it. And we can wait. We can say, okay, please go home, wait. Let's find it, what it is. If we do acid-fast stain, we find it's presumptive positive Mycobacteria tuberculosis, we have to do something. We have to use antibiotics. So, let's give you some examples. Some bacteria grow short, some bacteria grow really quick. So that's a generation time. Now, we're going to come out with some of the mass. We want to explain to you what the story behind it, or what it is. So here is the thing. I have N0. This means original bacterial cell. Original cell number. Okay, just simple. And I have NT. This means after time T, the bacterial cell. Which means how many bacteria are already there? Now, of course, we have a T. That means the time. Okay? So, what is going to be the formula looks like? Now, of course, we have another one, it's N. Okay, that's a generation. Generation of at time T, or we say after time T. So what it looks like, this formula is NT equals N0 multiplied by 2 times N. Then we have to transfer them to the log. So this for the log, usually log 10.